In this video, I will be showing you how to prepare smears from Slant cultures. The tools that you will need for this experiment include a china marker, one frosted glass slide, plastic pipette dropper, inoculating loop, one test tube, a Bunsen burner, germicide, Bacterial incinerator, paper towels, gloves, goggles, lab coat, Slant culture, slide box, and a biohazardous sharps container. First, you want to place a small drop of water on the center of the slide, almost like a half drop. To do this, we are going to be using a plastic pipette dropper. In lab, we would generally use a dropper bottle of distilled water, but because I don't have that available, we're just going to use this. Now put the small drop of water in the center of the slide. We don't want a lot of water because this slide has to air dry. The more water on the slide, the longer it's going to take for it to air dry. Our next step is to obtain a small amount of culture from the test tube. But before we do that, we must sterilize the inoculation loop using the Bacterial incinerator. After sterilizing the inoculation loop, take a small amount of culture from the test tube using your inoculating loop. Here's your culture. Just going to take the cap off and go in and scrape some of that out. Now, place the collected cells onto the drop of water on the slide. Slowly and gently mix the cells with the drop of water. You want to evenly distribute the culture onto the slide surface. The thinner the smear, the faster it will air dry and the better we separate the bacteria. After placing the collected cells onto the drop of water on the slide, re-incinerate the loop. This will burn off any remaining bacteria. Now let the slide completely air dry. If you do not allow it to completely dry, the arrangement of bacterial cells may be disrupted. Once the slide has completely air dried, heat fix the smear using the Bunsen burner. Make sure that the side with the bacterial cells and label is facing opposite of the direct flame. You want to heat the bottom of the slide. To properly heat fix the smear, you want to pass the slide just through the top of the blue flame of the Bunsen burner at least 3 times. We heat fix the smear so that the bacteria sticks to the slide. After heat fixing the smear, let the slide cool off. Once the slide is cooled off, it is ready to be stained. But until we are ready to stain the slide, store the slide in the slide box to keep it safe. Now, decontaminate yourself and your workspace. First, dispose of your gloves. You want to put these in the biohazardous waste container. Then use germicide to wash up your workspace. Once you've done that, use the paper towels to wipe down your workspace.